I think it's pretty solid advice.
Groups of cells in different parts of your brain, they're all connected.
The statistics just don't back it up. There's nothing here.
Any complex life you find has mitochondria.
We have discovered things from athletes juicing.
My name is Matt Kaberlein, and welcome to the Optispan podcast. All right. Hey, everyone. Welcome to the Optispan podcast. And today I want to do just a really brief touch on a paper that came across my radar recently. The title of this paper is Association Between Plant and Animal Protein and Biological Aging, Findings from the UK Biobank. And it was published in the European Journal of Nutrition. This is a paper from a group of scientists from Harbin Medical University in Harbin, China, which I've never heard of before. And the only reason that I even really took a look at this paper was because Simon Hill tweeted at myself and Charlie Brenner the title of this paper, along with one of the graphics from the paper with the quote, higher plant protein intake is inversely associated with biological aging, which, you know, obviously, given that I'm interested in biological aging and protein, I thought was an interesting conclusion from the paper. So I wanted to take a look, and now I'm going to share with you sort of my, again, high-level thoughts after having read the paper and sort of try to put it into context of what else we know. So I'd say the take-homes here are, first, I guess I'd qualify this as sort of mediocre science. Like it's pretty standard, kind of pedestrian, epidemiological, correlative stuff. There's nothing wrong, per se, with what they did, except for probably the entire interpretation. But I would also say the answer was pretty obvious going in. And in some ways, this appears designed to get an obvious result and perhaps push a narrative or an overinterpretation to fit an agenda that the authors wanted to push. So again, I'd say it's sort of mediocre science with terrible science communication. And I really do have to sort of question the review and editorial process when I see papers like this. And I'll kind of explain to you why I say that. So first of all, what did they do? This is data from the UK Biobank. UK Biobank is this enormous biomedical database. I think it's got now more than 500,000 people in the UK, collecting a whole bunch of different types of data, including blood biomarkers, some imaging, survey, genetics. This is like a super valuable resource. We really should have something similar to this in the United States, made available to researchers worldwide, which is great, but also comes, I think, with some downsides. It's a little bit of a double-edged sword. In many ways, this has seeded a ton of really interesting research. I've covered a couple of papers that came out of UK Biobank data on the podcast before. But unfortunately, it's also seeded a lot of really crappy research. So I'd say it's very much a mixed bag. The fact that anybody, regardless of their expertise, can get access and dive into this data and publish papers, which is really where I think the problem comes in with the lack of quality control in the publishing world these days, creates a very sort of uneven situation where it makes a lot of noise in many ways in the scientific literature. But the idea behind the UK Biobank, is great. And I completely support the idea of creating these large data sets and making them available to the world. So in this particular case, what they did was they took a subset of people from the UK Biobank, and they really looked at a couple of different types of data. They looked at food recall questionnaires that the participants completed about what they had eaten over the past several years. I don't really need to say anything more about that, except everyone probably is, and if you're not, should be aware of the limitations of these sorts of questionnaires. And then they used what they claim are 4 different assessments of biological age. And again, this is one of the issues I have with the paper. These are, assessments of, at least to some extent, health status, but they call them biological age measures. And my biggest criticism here is they kind of just made this up. Like the definition of their definition of biological age, as far as I know, has never been used in the literature before in this way. And that, I believe, is a little bit problematic, especially when that's in the title of the paper. So, they say specifically in the paper, there's a paragraph in the introduction that I thought was pretty interesting and probably worth discussing in a little bit more detail. They start with this comment that a single biomarker of aging may not systematically reflect true organismal aging. Absolutely. I agree 100%. Then they say biological age is a more ideal indicator as it uses a variety of biomarkers that accurately reflect the functioning of multiple organ systems and the health status of an individual to describe biological aging. So this is where it's a little bit problematic. So yeah, I guess theoretically biological age is a more ideal indicator of organismal aging. That's kind of self-evident. And it utilizes a variety of biomarkers that accurately reflect the functioning of organ systems and health status. Yes, that's true. Again, the problem is, what are those biomarkers and how do we measure them, and how do we decide which subset of biomarkers is a true reflection of biological aging? And I think importantly, how does that differ from health status? In other words, biological age certainly will be strongly associated with health status, given that health generally declines with biological age, but it's not a perfect relationship. And you can be unhealthy without having any change in your biological age. So this is where I think things start to get a little bit complicated. And really, I think what they're looking at here are quasi measures of health status in some ways, but that are not really validated to measure biological aging. So it's a little bit of, sloppy thinking, I think. So they go on and say earlier studies have proposed and validated multiple approaches to calculating biological age. That's half true. Earlier studies have proposed multiple approaches to calculating biological age. I would argue none have been really validated. I guess it depends on what would you need to see to have a validated marker of biological age. But certainly there's none that have universal consensus or even really, I think, broad consensus. There's this perception that the epigenetic clocks have achieved broad consensus in the field as markers of biological age. I think that's actually not true. I think when you actually talk to a diverse group of scientists in the field, there's actually very little consensus that epigenetic clocks are really measuring biological age. It just gets sort of presented that way by the people who want you to believe that, and because these clocks have started to be sold commercially and in some cases used by functional medicine providers and things like that. But there really is not a broad consensus that epigenetic clocks are measuring biological age within the scientific community. So that statement's half true. Many people have proposed different ways to measure biological age. None of them are really truly validated, and in the sense that there's broad consensus. Then they go on to say, such as, so these are the specific things that they use here, the Clamara-Duval method biological age and the PhenoAge algorithm, which can quantify biological age and predict mortality through commonly assessed clinical parameters. Okay. Allostatic load is also a key indicator of biological aging, reflecting the accumulated burden of chronic stress, and is associated with subsequent functional decline and death. So what I think is interesting here is they're not actually using any of the epigenetic clocks. These are blood-based clinical biomarker-based tools that they are using to assess what they're calling biological age. With the exception of PhenoAge, none of these are really broadly used in the biology of aging community as metrics of biological aging. Then they go on to say repetitive DNA sequences known as telomeres situated at the termini of eukaryotic chromosomes undergo attrition with each division of a somatic cell. So that's the fourth method that they're using, is telomere length. Telomere length has emerged as a potential biomarker of biological age, as shorter telomeres are associated with higher biological age. This has been well known for decades now, that telomeres shorten with age. Again, essentially zero consensus in the field as to whether or not telomere length is a useful measure of biological age. You'll find in this case, even a much smaller percentage than the epigenetic clock, much smaller percentage of people who will say that telomeres are a good metric of biological age, and I think a much larger proportion of people who have questions about that. One of the real challenges, as an aside, of using telomere length as a measure of biological age is that there aren't great technologies for measuring telomere length right now. So anyways, they use these 4 different assessments of health status, I would say none of which are validated for use in this way as a biological age metric. And they said, we're going to use all 4 of these, and together that we're just going to make up this new definition of biological age. So here's, again, where my biggest problem is. That's not scientifically rigorous. Like, it's just not okay to make up a new definition for biological age. Unfortunately, this seems to be the norm these days. And I would say this is really a larger failing of the community. Again, the epigenetic people were the first to kind of push this false equivalency. But now it seems like anybody can just willy-nilly decide, this is how I'm going to define biological age, and publish a paper. And reviewers, editors are not doing their jobs and allowing this to make it into the literature. And so this just creates, I think, a lot of confusion and noise. And at this point, I kind of feel like the term biological age is essentially meaningless at this point in the scientific literature because everybody's using it differently, which is unfortunate, and I think in some ways reflects poorly on the reputation and quality of the field as a whole. So that's kind of where we are. But in this specific case, again, they're using allostatic load, which has been around since 1993. This Clamara-Duval method, which has been around since 2006. Again, I've never really seen anybody in the biological aging literature use this method. PhenoAge, which has an epigenetic correlate, right? So this is work from Morgan Levine, but there's also a clinical chemistry, blood chemistry method you can use to calculate PhenoAge, and then telomere length. So those are the 4 methods used here. All come from blood. And again, I think all are telling us something about health status, something about biological age. Certainly, I think in the case of PhenoAge and telomere length, you can make a pretty good case that these are reflecting aspects of the hallmarks of aging at least. But I think clearly all are reflecting some aspect of health. I actually think it's kind of funny. I went and looked at the Clamara-Duval paper from 2006. This sentence from the abstract I thought was just a little bit interesting given where the field's at today. So they actually say in the abstract of that paper, the lack of exact definition of the concept of biological age is a typical feature of works concerning biological age. Again, this was back in 2006 before the clocks had even really been talked about. That is why comparison of results of various published methods makes little sense, and eventual proof of their optimality is impossible. Couldn't have said it better myself. Okay. Unfortunately, we haven't really changed since 2006 in that way. Let's move on to the main findings of the paper. The main findings, I would say, are that higher plant protein intake was associated with better scores on these 4 tests that they used. In other words, if we were to accept those are tests of biological age, higher intake of plant protein would be associated with lower biological age. If we accept their measures of health status, would be associated with better health status. Animal protein did not have any significant or strong effect. Okay, so plant protein intake, better health based on these measures. Animal protein, no significant effect. But processed meat had a negative relationship. Again, nothing surprising, I think, there, that processed meat is not good for you. One interesting thing I thought was that the relationship between plant protein and better scores by these metrics seemed to be at least somewhat, if not largely, driven by 3 specific blood parameters: alanine transaminase, aspartate transaminase, and gamma-glutamyl transferase. These are all measures of liver function. And so, one reasonable interpretation is that what's driving these better health scores or health-associated scores is really either an improvement in liver function or more likely a reduction in liver disease among the people who are eating more plant protein. I think that's a very plausible explanation for the results that we're seeing here. And then that's basically it. And then there was a bunch of what I would consider pretty speculative discussion, mostly trying to create potentially causal explanations for correlative observations. And so again, I think the most likely explanation for everything here is really that people who eat more plant protein, yogurt, nuts, are generally eating fresh foods, and they're eating less ultra-processed foods. And we know ultra-processed foods and foods that have a lot of simple sugars supplemented are bad for you. And so if you're eating less of those things and eating more fresh food and plants, you're probably going to have better health. That, I think, is the most likely explanation for the relationship here. I'm guessing it has essentially nothing to do with the plant protein itself and has more to do with the overall diet quality. Likewise, people who tend to eat processed meat tend to eat more processed other stuff, which we know is bad for you. And so I think that probably accounts for the association between higher levels of processed meat and the lower scores on these tests and potentially poorer liver function. Again, that's an alternative explanation. And I do think it's important to appreciate that just because it's an alternative explanation that can explain the data doesn't mean it's right. I think it's probably right because we know of lots of other cases where similar sorts of relationships have been seen. I think that's a much more likely explanation than that the source of the protein itself has anything to do with anything in this context. I personally find it extremely unlikely that plant protein versus animal protein has any big differences in health, period. Like, just don't think there's any good reason to believe that. Yes, it's absolutely possible that there are incremental, tiny effects based on the source of the protein itself. But again, I think it's much more likely that the context of what else you are eating matters much, 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 much more. So, folks, don't major in the minor. And unfortunately, the title of this paper makes it seem like it's the plant protein versus the meat protein that's important. Again, that seems very unlikely, given the way the study was done and given everything else that we know. So again, back to the original tweet. The comment section here was pretty much a train wreck that I'm really not going to wade into. And I do have to say, while it is a statistical certainty that 50% of the population is below average, it kind of feels like that distribution has gotten skewed on X over the last 16, 18 months or so. I do want to just offer a few final reflections. First, I've said it before, sure I'll say it again, just ignore the people who speak with 100% confidence about interpreting studies like this or about what sort of diet is optimal for you. I think there are some general themes, but there really isn't a single diet that is optimal for everyone. Second, I'm not 100% certain, but I'm 98.36% sure that neither carnivore nor vegan are optimal for most people. Okay, I think those are both extremes. And like many things in life, extremes in nutrition very rarely going to get you where you want to be. Third, when it comes to diet, I think the sort of general principles are shop around the edge of the grocery store, eat mostly fresh or minimally processed foods, limit alcohol intake, avoid added sugar whenever possible. You do that, you're going to be better off than 95% of the people out there. And I think that's the take home from this study. That probably can account for much of the results that we're seeing in this study. Do the people eating a healthier diet, which might tend to be higher in plant protein, have a slower rate of biological aging? Maybe, probably. Do these tests that they used here show that? I don't think so, because I don't think that those tests are really measuring biological aging. I think they are telling us something about health, which is telling us something about biological age. So they're connected, but I certainly wouldn't say that this definitively demonstrates a reduction in the rate of biological aging in the people who ate a diet that was higher in plant protein and probably also was an overall healthier diet than the other people. Okay, so that's it. Hopefully that is helpful, useful, at least can make you think about some of the nuances and some of the ways that this stuff gets misrepresented in the scientific literature and on social media. As always, if you have got any comments or questions, please leave them below. If you like this video, hit that like button. I hope you subscribe and hope to see you next time on the podcast.